Hello students, now we will start with a new topic that is about enzymes. What are enzymes? All enzymes are proteins, nothing but. They are the deviated forms of the tertiary structure of the proteins. These enzymes attain an special nature of catalyzing the reactions. So the enzymes that are present in living organisms are also termed as biocatalysts. So enzymes are biocatalysts in living organism body. Enzymes are proteins, means all enzymes are proteins. Now, enzymes are synthesized at tertiary structure of the protein. Enzymes are termed by Kuhne. Term is given by Kuhne. Next, the first extracted enzyme is gymase from the yeast cells by Buchner. First extracted enzyme is gymase from yeast cells by Buchner. Now, there are different types of enzymes. We will see some of the examples. Some enzyme or some RNA behave as enzymes. Some types of RNA fragments, they behave as enzymes called as ribozymes. Now, enzymes have active sites for substrate binding, during reactions. Next, enzymes are affected by temperature, pH and concentration of the substrates. If we see the first one, temperature. Enzymes are very much active at optimum temperature, that is 25 to 40 to 45 degrees centigrade of temperature. If the temperature is more than that, then enzymes get denatured, means they will get broken down. If the temperature is very less, then enzymes are considered to be inactive. But there are some exceptions, like Taq polymerase is one of the enzyme that is still active at 80 to 90 degrees centigrade of temperature also. So, enzymes at above 45 degrees centigrade are denatured. enzymes between 25 degrees centigrade to 45 degrees centigrade are active in function. Next, enzymes below 25 degrees centigrade are inactive. But here there is an exception, which you read in biotechnology application part. During the PCR technology, you will read about this Taq polymerase. During the extension step, exception is Taq polymerase, which is active at 80 degrees centigrade to 90 degrees centigrade also. This Taq polymerase is also called as DNA polymerase of Thermophilus aquaticus organism. What is use of enzymes? Living organisms' body is having chemicals. Can't the chemicals carry on the reactions? What is need of enzymes to be involved in the reactions that are happening inside the living organism's body? Chemicals are termed as inorganic catalysts. Now, the basic nature of an inorganic catalyst is to get expressed in two different forms. That is, they will undergo physical changes and chemical changes. Chemicals, majorly, they will show two types of changes or forms, that is physical form and chemical form. They also undergo physical changes and chemical changes. Under example of physical changes, ice, which is solid in nature, it is getting converted to water, which is liquid form. Coming to the chemical changes, here by breaking the bonds, the energy is released, but the energy is very less in amount. That is not sufficient for the living organisms to survive. Here, energy released by breaking the bonds is very less or insufficient for organism to survive. Here we can take examples like starch converting into glucose. Here we can see one point, like glucose again still has to get broken down to release energy. It is a long process. One more example is barium hydroxide. When it is fused with sulfuric acid, it gives out barium sulfate plus 2 water molecules. Still the energy released here is less. But this is not happening inside living organisms' body, then why we are reading here? If we consider this one as one best example for chemical reaction, here it needs more temperature to forward the reaction or for the completion of reaction. Here, more temperature is required. But we know that living organism's body cannot produce means more heat in their body. If it is so, organisms cannot survive. Since more temperature is required and that is not provided by the living organisms, chemical changes as it is cannot happen in the living organism's body. Then what about starch? It is getting converted into glucose in animal body and plant body. Yes. In case of this starch and glucose, if we add water molecules, it is enough. There is no need of high temperature. But the negative thing about this chemical reaction is glucose, again it has to involve in the total aerobic respiration or anaerobic respiration. And in case of aerobic respiration, it has to release out 38 ATPs. And in anaerobic respiration, it has to give out only 2 ATPs. Since here again addition of molecule and time being is more, this is also not considered as the best chemical reaction that can be means only fit in the living organism's body. Means still we need some of the changes in the chemical reactions. Now living organisms have adopted the involvement of enzymes in the metabolic activities or reactions to release energy quickly and in a short time with less temperature conditions. Vary very more for every change of 10 degrees centigrade. For change of every 10 degrees centigrade. It means that whenever in case of temperature changes are there, means 10 degrees increased or 10 degrees decreased, then the mode of chemical reaction and the products obtained, they are very much showing drastic results. Different types of results are being obtained. So some imbalance happens during metabolic reactions. To balance those imbalanced type of reactions, enzymes are involved in activities. To balance those reactions, enzymes are involved in metabolisms. We will see one example regarding the slow reaction and also the fast reactions. Just one example we will see. CO2 when binded with water in absence of enzyme, it gives carbonic acid. In absence of enzyme, this is considered as slow reaction. Here, if we apply only 200 number of CO2 molecules, 200 number of water molecules, we get 200 number of carbonic acid. It is okay. But in a second or when we put some time limit for this, it happens very slowly and it takes more time than the limited time period. The same CO2, same water molecule, when in presence of enzyme, it will give carbonic acid normally it will give, but we are seeing about presence or absence of enzymes. It will give nearly 6 lakhs of CO2 molecules, 6 lakhs of water molecules, will give 6 lakhs of carbonic acid molecules in a second. In a second, we are getting nearly 6 lakh. The same thing means the quantity of the enzyme and the product is not seen in absence of the enzymes. Now, again, in the next second, again in a second, this carbonic acid can again be dissociated into CO2 plus water in the lungs. Means within 2 seconds the total reaction is being completed. Under use of enzymes, we have explained some concept that it increases the reaction rate and also decreases the time that is required for completion of reaction. So it increases the reaction rate and decreases the time required for reaction. Next thing, enzymes have active sites for substrates to bind on them and forward the reaction for substrates. In the same way, substrates have some energy that they have to spend during the involvement of the reactions. So substrates have some energy to involve in reactions. We call that energy as activation energy. We can write one complete enzyme plus substrate involved reaction as enzyme plus substrate gives enzyme substrate complex, which later gives the product, and enzymes get separated from this complexly binded form. Now enzymes are free. Now they can again bind with other substrates and carry on the reactions. There are some types of reactions, metabolic reactions, that need many enzymes. Some metabolic reactions need many enzymes to complete the reaction. Example we can take as glycolysis. Glycolysis, where glucose is getting converted into pyruvic acid, and here nearly 10 steps are involved and more than 10 number of enzymes. We can take 10 number of enzymes approximately are involved to complete the reactions. Told you that substrates need to spend some of the energy that is called as activation energy. Basing on the activation energy in presence of the enzyme and in absence of the enzyme, how substrates are involving in the reaction. Let's plot a graph. This is x-axis and this is y-axis. Take out a substrate that is involving in the reaction. If it has to complete the complete reaction and give out products, it will take, means it has to spend more energy. Activation energy required to give out the products is more. So this long line, it is indicating activation energy. Now, when we add enzymes to the substrates, enzyme plus substrate in the second case, then substrate needs to spend very less of its activation energy to give out products, because enzyme is catabolizing the reaction as we have seen before some of the examples like CO2 plus water. Now this level indicates less activation energy spent by substrates. Now, the level where enzyme substrate complex has formed, that is considered as by the time total active sites of the enzymes are occupied by the substrates, and there whatever the energy that is required by the substrate is finished. There is no more activation energy to involve in the reaction. So this is termed as transition state. Transition state is maximum energy required by the substrates to involve in reactions, to involve or participate in reactions. By the time of enzyme substrate complex, all active sites of enzymes so here we cannot write this enzyme. So transition state is meant for only enzyme substrates. This. Here we cannot write enzyme. By mistake I have written here, because this black color graph we are representing by the energy spent by only substrates. There is no point of enzyme inclusion here. But in the second case, enzyme plus substrate both are involving. So we have to mention here enzyme substrate complex are occupied by substrates. So this is about the graph that is representing based on the transition state and also activation energy that is spent by the substrates to involve in the reaction. In first case, in this black color graph, there is no point of involvement of enzyme, so that substrates have spent more activation energy to give out products. But in the second case, there is involvement of enzyme, so substrate can spend very less energy. The remaining forwarded reaction will be catabolized by the enzymes that are present. Next is factors affecting enzymatic activity. Temperature. Temperature we see. both in case of temperature and pH, the factors that affect the enzymatic action, the graph looks same. Like this. On x-axis, we take here temperature, and on y-axis, we take enzymatic activity. Here also the same, enzyme activity. But here on x-axis, we take pH. First we will see about temperature. As I have told you before, enzymes are active between 25 to 40 to 45 degrees of temperature, but they get denatured above the 45 degrees of temperature and are inactive below 25 degrees of temperature. Then what about the temperature, or in case of human body? We have 36 to 37 degrees of temperature. In the graph, if you see, this point has enzyme substrate complex, or this is considered as the maximum active condition of the enzyme. So by the time of 40 degrees centigrade, enzyme is very much active to give out the products. Fine. But human body, this is considered as maximum temperature or optimal temperature, where enzymes are actively participating in the reaction. But in case of humans, the temperature is 36 degrees to 37 degrees centigrade. This point is called as suboptimal temperature. Next, different enzymes will be acting at different temperatures, but exception already we have discussed. I will tell you, remind one more time. Exception is Taq polymerase. It is active at 80 degrees centigrade to 90 degrees centigrade of temperature, and this is extracted from Thermophilus aquaticus bacteria, next, this is also a DNA polymerase. I already told you, Taq polymerase we read when we are reading about PCR technology. In the third step, that is extension step, we use Taq polymerase enzyme. pH. There are many secretions in our body, digestive enzymes are there. All of them are having different types of pHs. So, in human digestive enzymes, we will see the pHs of different types of enzymes. One is salivary amylase. Salivary amylase, also called as ptyalin. Its pH is 6.8, near to the neutral. Next, tryptophan will take, which is secreted in the stomach. Its pH is acidic, that is 1.2 to 1.8. Next, another one, trypsin, which is secreted in the pancreas. Its pH is basic, 8.6 to 8.8. So, all enzymes are not having same pH levels for their activation. Different enzymes will be having different pH levels. So, this pH level, enzyme is active at different pH levels. So, though the graph is in case of one enzyme it is like this, it depends. We can write the values. It can be changed from like this, basing on the type of enzyme and their pH levels. But finally, the graph for temperature and pH levels is same regarding the enzymatic activity. Here, as when initially the enzymes plus substrates are involved in the reaction will be forwarded very normally. But by the time enzyme substrate has complex has formed and all the active sites of the enzymes are completely filled by the substrates, then there is no more increasing of the reaction. Then later, after formation of enzyme substrate complex, the only next step is giving out products. As enzymes plus substrates are involved in a reaction, at a point, all active sites of enzymes will be occupied by substrates that leads to only formation of products but not more accumulation of enzyme substrate complexes. Immediately, when enzyme substrate complex has formed, it is readily involving in the next step of giving out the products. If we plot a graph like this and give out substrate concentration on x-axis and rate of reaction on y-axis, the graph will be represented like this. Means this point we can consider as enzyme substrate complex. And here there are different terms to be given. This is called steady state of the graph. This is called as lag phase of the reaction. This is called exponential phase or log phase of the reaction. Means here the activity of enzyme plus substrate is very high and very accurate and very perfect during the exponential phase. But after some time, all the enzymes are active sites are occupied by the substrate. So there is no more existence of forwarding the reaction in a straight linear way like this. So the enzyme comes to a steady state or the plateau. So this one is also termed as plateau. Later, the same enzyme substrate concentration based on increasing and decreasing levels of substrate levels, Michaelis-Menten constant was given. Next we will see about mechanism of enzymatic action. There are two different mechanisms. One is lock and key mechanism, which is given by Fischer. In this we see one enzyme when it is involved with a substrate. So this one we can consider as an enzyme and this one as an substrates. Since here I have represented two different active sites, we will give two different substrates. This is S1 and S2. When they are involved in the reaction, this enzyme in the next step, it will be binded with substrate 2 and substrate 1 When enzyme substrate complex has formed, then at that stage a bond will be formed between enzymes between the two substrates, not between enzyme and substrate. Substrates are fitted in the active sites and there will be a bond formation. In the next step, these bond formed S1 and S2 will be giving out the product and enzyme gets separated as that of previously. So enzyme plus now the product has been formed. There is no existence of substrate 1 and substrate 2 now because a bond is responsible for converting their form, physical form, into products. So this will be product. Now enzyme is ready again to bind with other types or number of substrates and again re-involve in the reactions. This was explained by Fischer. Next, second mechanism is induced fit model given by Koshland. Induced fit mechanism or model. This is given by Koshland. Now here, active sites are normally present in case of enzymes. Those sites are of two different types in some enzymes. That concept is given by Koshland. He said that active sites are of two types. One is catalytic site or catalytic site. Next is buttressing site. So what is function of catalytic site? Its function is to break the old bonds and frame out new bonds. Its function is to break old bonds and frame new bonds. Next, buttressing site. Its function is to provide space, means completely working as the active sites only for attachment of substrate. Behaves purely as active sites for substrate binding. Consider the enzyme like this representing two different sites. We can consider this one as catalytic site or catalytic site, and here the place as buttressing site. Now when two different types of substrates have been entered to involve in the reaction, S1 and S2, then in the next step, buttressing site will be same, it will not be changing its position, but now catalytic site is changing its position to fit the substrates firmly into the buttressing site active places. So here catalytic site has now bent to give firmness for the substrate. This is S1 and this S2. Now S1 and S2 can't move anywhere because those are fitted by catalytic site due to its bending nature and pressing towards the active sites. Here forms the bond between S1 and S2 that later is responsible for giving out the products. We can also call it as enzyme substrate complex. In the next step, again enzyme will be coming to its original position. This is buttressing site. And this is catalytic site. We are getting now the products. This method is explained by Koshland. Next, about competitive and non-competitive inhibitions. So first one, competitive inhibition. If you see the first meaning of this, nothing but some inhibitors along with the substrates are occupying the active sites and preventing nearly half or some number of active sites, occupying some number of active sites and not allowing the substrates to bind there, so that we are not getting 100% of the products. So there is a competition for the active site between the inhibitor and the substrate. So under competitive inhibition, there is competition for the active site and that competition is between inhibitor and substrate. So this one also we will explain in the form of a diagrammatic representation. So this is one active site, this is another active site. So what happened now? Inhibitor has come and binded to one active site. In the next case, substrate has come and it is trying to fit here, right, equally. So this is occupied by inhibitor and this active site is occupied by substrate. So there is a competition between inhibitor and substrate for the active sites. In the next case, we are getting enzyme substrate complex, but here there is no bond between enzymes and inhibitor and substrate. So we are getting 50% of enzyme inhibitor complex and 50% of enzyme substrate complex. Here we are seeing only 50% of products. We are not getting 100% of the total products. It also means that 50% of the active sites are occupied by the inhibitor. Now, what examples can we take to explain? In Krebs cycle, in one step, we will see succinate getting converted into fumarate in presence of enzyme succinate dehydrogenase. Now, this is the substrate that has to bind to the active site of succinate dehydrogenase. There is an inhibitor called as malonate. Malonate. This malonate is inhibitor here. It again binds to some of the active sites of the enzyme and stops 100% formation of the fumarate. So in case of Krebs cycle, this is one of the best example to explain competitive inhibition. Another example we can take, second example as, this is one example, second. Para-amino-benzoic acid in presence of enzyme folic acid synthase. Folic acid synthase it gives folic acid. But this normally happens in animal body. But what is happening? There is an inhibitor called as sulfa drug, nothing but the painkillers. Sulfa drug. Here, this is the inhibitor. It also binds to some of the active sites of folic acid synthase enzyme, so that we are not getting 100% of folic acid synthesis in the body. This is another example. So under competitive inhibition, we have seen two examples. One in Krebs cycle, another is because of sulfa drugs. What is full form of PABA? Para-amino benzoic acid. PABA means para-amino benzoic acid, precursor for the formation of folic acid. Here, again inhibitors are trying to involve in the reaction, but they are not binding at the active sites. They are binding at other place other than the active site and changing the shapes of active sites, so that even 50% of the substrate also couldn't bind to the active sites. Here, inhibitor, non-competitive inhibitor binds on enzyme at a place other than active sites. And changes shape of active site. Shape of active sites. If we diagrammatically represent this one, so here will be the substrate S1, other substrate S2. They are ready to fit here. But there is another non-competitive inhibitor. For suppose think that shape is like this, it will come and bind to other place of the enzyme. So in the next step, it can be represented as like this. When this non-competitive inhibitor has binded to some other place on the enzyme, then the active sites shapes are being changed like this. But enzyme substrate S1 and S2 are like this. So they cannot fit into the active sites, so that 100% of the products are not being formed. So no active site for substrates. So no product formation. Here also we can take one best example like cyanide. Cyanide is non-competitive inhibitor. It binds to cytochrome oxidase, the fourth complex in electron transport system, where one water molecule is formed for every NADH or FADH breakdown. Cytochrome oxidase. Another example, penicillin. Penicillin, it stops the formation of peptidoglycan membrane or the cell wall formation in bacteria. So this is one example. This is another example. Stops cell wall formation in bacteria. So this is about competitive and non-competitive inhibition. One more inhibition is there, that is allosteric inhibition. Allosteric inhibition. It is also called as feedback inhibition. Here we can take example like glucose in presence of enzyme hexokinase giving out glucose 6-phosphate with help of ATP. But here, if body doesn't require more number of glucose 6-phosphates, then the glucose 6-phosphate itself will come and bind to hexokinase and it will stop the forwarding reaction. So, it goes back and stops hexokinase from forwarding the reaction. So this is one of the best example for feedback inhibition or allosteric inhibition.